So in the last segment, we were looking at monohybrid crosses, and fundamentally a monohybrid cross is a cross between two hybrids that are hybrid for the same gene. So we did big little p, cross big P, little p. That's a monohybrid cross. In our modern language, when you have two alleles that are different, we don't say hybrid, we say heterozygous. But Mendel said hybrid. Now we're going to look at what's called a dihybrid cross. A monohybrid is just a hybrid or a heterozygous for one gene. So we did flower color in the last section. A dihybrid cross, you would start with a cross. The cross is between two heterozygotes that are heterozygous for two genes of the same between the two organisms. So an easier way to say that is down here at the bottom. It says the dihybrid cross is a cross, always a cross between two double heterozygotes. Here I'm using the letters big R, big Y, little y. It could be any letter combinations, but two double heterozygous individuals. Or it could be a self-fertilization involving one individual who's acting both as the mother and the father. Now, the way you get a dihybrid individual is the same way that you get any hybrid individual. You start by crossing the true breeding P1 plants. And what Mendel did was he had plants that were true breeding for two traits. So big R, big Y, little r, little y, little y. What this means is the R gene, whether it's a big R or a little r version, the R gene is on one homologous pair of chromosomes, and the Y gene is on a different homologous pair. So I usually like to think of, for example, the R gene, and the first gene doesn't have to be the letter R, but whatever gene is in the first position here of this genotype, that is, if I draw a homologous pair of chromosomes, then in this cell, both of them have a big R. And in the same cell, I have a small pair of chromosomes that each have a big Y allele. Big Y, big Y. And that represents every cell in that parent. Now, in this other parent, every cell would have, on the big pair of chromosomes, they would have a little r on both. And on the small pair, they would have a little y on both. So they're just saying, like, let's say you started with this. So all the cells are big R, and little y. Sorry, little r, and little y. And all the cells in this other parent are big R, big Y, big Y. But anytime you have two letters of the same type, like R, two R's, that's a homologous pair. And anytime you have two Y's, that's a different homologous pair. And so you want to think of these letter combinations in terms of homologous pairs in each cell of that organism. And this one as homologous pairs in each cell of the organism. And so for thinking about this chromosomally, every gamete from this parent has to have a big chromosome with a big R and a little chromosome with a big Y. Every gamete from this parent has to have a big chromosome with a little R and a little chromosome with a little Y. And so when those fuse together, there's really only one way you can get. You get the big R from this parent and the big Y from this parent. You get the little r and the little y, but you don't need a Punnett square for this. There's only one way this can shake out. But what gets interesting is when you cross two of these individuals to each other, then you do have a bunch of possibilities that you could get in the offspring. And so when the F1 self-fertilizes, it's like crossing it to itself. It can be represented this way: big R, big Y, little y, cross to big R, big Y, little y. And I'm going to show you the Punnett square, but you can basically get all kinds of combinations in the offspring, which are the F2 offspring. And the F2 plants then will fall into this pattern. Out of 16 possible combinations, 9 of them will follow this genetic pattern, meaning they'll have one big R, and then the blank means this could be a big R or a little r. And they will also have one big Y, and the blank means this could be a big Y or a little y. And when I use this shorthand, what I'm saying is that 9 out of 16 offspring are going to have round, because big R is round, yellow seeds. Yellow is dominant too. Big Y is yellow. 3 out of 16, out of every 16 offspring, 3 will be plants that make round seeds, but the seeds are not only round but green, because green is the recessive, green is the little y. 3 out of 16 are sort of the flip side. They'll have wrinkled seeds that are yellow. So if you look at this pattern here, these two groups are like exact opposites. Each of them is dominant for one trait and recessive for the other trait in opposite ways. And then one offspring from this cross out of 16 would be recessive for shape, wrinkled, and recessive for color, so green. But this is your pattern every time you do a dihybrid cross, which the parents would have to follow this pattern. Now, the letters can be different. If you have big A, big B, little b, cross, big A, big B, little b, that's a dihybrid cross. It's just different letters. But you can choose what letters you want to use. Remember, that's a shorthand for what alleles are on the chromosomes. So the letters represent the alleles on the chromosomes. The chromosomes are segregated in meiosis, such that each gamete is haploid, and then 2 haploid gametes come back together to make a new offspring. And so you can get all kinds of combinations with a dihybrid cross, which is what we're showing here. So this is your textbook, says thinking about a dihybrid cross from a chromosomal perspective. I'm really happy they created this concept. I've been doing a lecture like this for a while. And so they're actually showing these alleles. The big R's are these blue alleles on the long pair of chromosomes. They're showing monads. And the big Y is on this short pair of chromosomes. And then the little r is the red stripe on the large chromosome, and the little y is the green stripe. Yeah, green stripe on the little chromosome. Meiosis will make only one kind of gamete from each of the P1 plants. But when you get here, you have this F1 that is big R, big Y, little y. And then when you self-fertilize that, that's when you get all kinds of offspring. So here's where they show it all. This is the Punnett square. So this is the chromosomal depiction of the gametes. So let's say this is an egg, this is an egg, this is an egg, and this is an egg. What you're seeing is that each egg has to have an R and a Y. But since the mother here has big R and little r, big Y and little y, you can get any combination in the egg. So there's 4 different slightly genetically different eggs. And since it's a self-fertilization, the father is the same. And so you can get 4 genetically different sperm types. And I put tails on my sperm. And then when any egg meets with any sperm, they fuse here and you get the genotype of the offspring. I don't like that they show a pea here. It's a plant is the offspring, and the plant makes peas. But the plant has this genetic makeup, this plant has this genetic makeup, and so on. But when you sort them all out, 9/16 of the plants will have round and yellow peas, 3/16 of the plants will have round and green peas, 3/16 of the plants will have wrinkled and yellow peas, 1/16 of the plants will have wrinkled and green peas. So that's your pattern again. But it doesn't have to be these particular genes, it's just the pattern. The dominant allele on both traits will show in 9 out of 16 offspring. A dominant trait and a recessive trait will show in 3 out of 16 of the offspring, and then the inverse way in the other 3 out of 16 offspring. And then 1 out of 16 offspring will be homozygous recessive for both traits. And so the dihybrid cross let Mendel track 2 genes at the same time, and that led to the law of independent assortment, which is the second law. And basically he's saying that if the seed color gene is on the long homologous pair of chromosomes and the seed shape gene is on the short homologous pair, then the way that they sort out is random, and you can get all the different combinations in the offspring. Now we know that the independent alignment of the homologous pairs during metaphase 1 leads to independent segregation of these different traits, such that a gamete can get an allele for round seeds and an allele for yellow seeds, or an allele for round seeds and an allele for green seeds. Those combinations are possible because these are on different chromosomes, different homologous pairs. All right, I will finish up with a little bit of probability and then some special cases that Mendel didn't know about in the next lecture segment.